this is going to be a lecture about lymphocyte development. A lot of these concepts will primarily overlap with immunology, so if you require any help in both immunology and hematology, then this will be a good lecture for you. The objectives here scan mostly because this is directed at my own students. I would recommend following along with the objectives listed in RODAC's Hematology Clinical Principles and Applications to see where you need to go in terms of developing your understanding of the material. So lymphocytes are agranular leukocytes. That means they are white cells that do not have any granules inside of them. They help play a role in protection against pathogens and malignancies. Um, if you look at a lymphocyte underneath the microscope in the peripheral smear, you will not be able to tell apart a T cell, a B cell, an NK cell, any T helper cells, or natural or killer cells. You will not be able to tell them apart. So when you look on your CBC or your manual differential, they will simply just be called lymphocytes. If a provider or a pathologist requires to know the percentage of the subtypes of each lymphocyte, then they will require flow cytometry or specialized stains to be able to tell them apart. T and B cells are part of your adaptive immune system. This involves a lot of immunological memory, humoral, and cell-mediated responses. Natural killer cells are part of your innate immune system, and they play a cytotoxic role, meaning they'll be able to get rid of anything that your body recognizes as non-self. The adaptive immune system takes a lot more time to develop, but when the response is able to be developed, that response is really good at being very specific, more strong, and more effective than the innate system is. Lymphopoiesis just means the development of lymphocytes. Lymphocytes will develop from pluripotent hematopoietic stem cells, which are located in your bone marrow. Lymphocytes come from the common lymphoid progenitor and will develop into T, B, and natural killer lymphocytes. Um, if if you are very, very nerdy, and you are I don't know what's happening here. Okay, that's fun. Um, if you're very nerdy and you enjoy immunology to a very extreme degree, then I would recommend looking at these transcriptors regulators and the cytokines to be able to better associate them with T and B lymphocytes. If you are looking at this as a med lab student, I'd be kind of shocked if the board asked you about these. T and B lymphopoiesis can be divided into two different phases. First is the antigen independent maturation process, and next is the antigen dependent maturation process. Antigen independent just means that they do not require antigen presentation at this stage. This occurs within your primary lymphoid tissues. So basically, all we're doing is we're just developing from this common lymphoid progenitor and ending up with cells that have either differentiated into T and B cells. But these T and B cells are immunocompetent, but they still are naive, meaning they haven't been associated with it. They haven't learned a specific antigen just yet. In order to learn a specific antigen, they have to migrate to the secondary organs. This is where antigen dependent maturation comes in, and this occurs within your secondary lymphoid tissues. So, what happens is that these immunocompetent but naive lymphocytes are exposed to both self and non self antigens, and by being able to recognize those, then they're able to continue developing during lymphopoiesis. We also talk a lot about these T cell receptors and these B cell receptors. The stars here just indicate which lineage that we're about to talk about, so let's talk about it. So most hematology CBCs especially, we prefer talking about the relative distribution rather than the absolute count. Relative distribution just means the absolute or the percentage amount found in the manual differential. If you require the absolute count, then I usually recommend just multiplying the percentage with the total white count. But this is still good for you to know. Just generally recognize um, this number as your absolute count. Until you are about 4 years old, lymphocytes will actually become the most abundant leukocyte, because your body is desperately trying to adapt, to the fact that you are now outside of mom in order to develop a proper immune system. Once your immune system becomes much more competent, then your neutrophils will start overtaking it. The fact when your lymphocytes overtake your neutrophils like this, we will call it an inverted differential. In an adult, lymphocytes take about 20 to 40% of all the white cells in your peripheral blood. Within that breakdown, we're looking at about 15 to 30% of those lymphocytes are B cells, about 5 to 15 are NK cells, and the majority of it being T cells. And within that T cell subgroup, we are looking at majority of the T cells being CD4 T helper cells. 95% of all lymphocytes will reside in your tissues, your spleen, your lymph node, and bone marrow. And 5% of all lymphocytes reside in your peripheral blood. Basically, what they're doing is they're just traveling from one tissue to another. Most lymphocytes will live for a very long time. And will have encountered their antigen at some point. Um, if not, if they're still naive, then they only exist for about 5 days. So in order for T and B cells to function, they have to interact with macrophages. Macrophages' job is to present these T and B lymphocytes with these foreign antigens. Upon this presentation, then that will immediately activate the adaptive immune response. In order to be able to regulate this entire process, then we require cytokines. Those cytokines are secreted by your T, B, natural killer cells, and macrophages. B cells' role in the adaptive immune system response is to produce antibody, or the humoral arm of the adaptive immunity. T cells' job is to kill off abnormal cells. This is the cell-mediated arm of adaptive immunity, and this will also include viral or cancerous cells. So this is just a really good presentation of what happens when you get the first response versus the second response. Notice that you have notice the amount of antibody that you are um, creating overall, especially at the peak of about 15 days or so. There's sort of a plateau that you notice in the total antibody. But upon the second time that your body has encountered a foreign antigen, that spike goes way up. That means your body is able to have a much more effective and rapid immune response versus your primary response. Leukocyte adhesion receptors are very important for your lymphocytes to be able to migrate between that blood and lymphatic tissue. They also require those receptors to be able to interact with other immune cells for cell signaling and activation. A lot of these receptors are integrins or immunoglobulins. The ones that are not bolded, you can memorize if you really wish, but the bolded ones is the ones that you're going to hear about most common. Those CD4 and CD8 markers, and that also helps us tell apart which type of T cell that we are looking for. Unfortunately, as you get older, immunocompetence will start to diminish. A lot of this is just due to your body just aging over time. When you're about 40 years old, your thymus has shrunk considerably from when you were a baby. So that means the thymus isn't able to support the immune functions as well as it used to. Also, your memory T cells are unable to replicate as effectively as they were when you were much younger, which leads to a weakened immune system. This is why you have to take good care to make sure, especially when you're in an elderly care setting, that you have to be aware that older patients are much more susceptible to not only just infections but also to cancers as well. Also, older people are much more prone to developing autoimmune disorders. Your body's ability to recognize antigens that are associated with self versus non-self gets worse. And when that happens, your body might be incorrectly inclined to try to attack anything that would be normally considered self. And that's where a lot of these autoimmune disorders occur. All right. If you are a rheumatologist, or you are studying immunology, unfortunately, you will have to know all this. But if you are a med lab scientist, I would say develop a general association, but don't torture yourself with everything here. Lymphocyte development. This starts with your VD and J gene arrangements. So lymphocytes have to be able to recognize recognition receptors to be able to recognize that specific or unique pathogen's antigen. In order to account for the near infinite amount of pathogens that we can encounter in the world, um, we have these rearrangements in your VD and J genes, and that can account for a large number of variations between your B cell and your T cell receptors. We also talk about the VD and J genes, developing those heavy chains that are associated with the IgGs that are found on B lymphocytes. So this is what VD and J arrangement looks like. Basically, what we're doing is that we're splicing these genes at random parts of the genes. Once we've spliced what we want and what we need, then that will lead into an IgM, or that will lead into this heavy chain protein being developed. This random number, if we are able to find a match with an antigen, then that VDJ arrangement plays a huge role in just getting the possibility of a non-self antigen being able to be recognized. So, just with this process, we can create billions of alleles. And because of that, that means the receptors are really both highly variable and pathogen specific. So the way that we sort of test whether or not the receptors works is that the lymphocytes have to be able to bind to the antigen with this unique combination of VDJ arrangements. When they are able to recognize it, then they start developing both phenotypically and functionally to create these different subsections that I keep talking about. Um, for your T cells, we have the cytotoxic T cells, whose job is to kill abnormal cells. Helper T cells, whose job is to help activate B cells, T cells, and macrophages, and regulatory T cells, who help suppress T cell immune responses. We also talk about within the B lymphocytes, we talk about B1 and B2. Again, I cannot stress this enough. If you're looking at a lymphocyte just from peripheral smear, you cannot tell it apart. If you need to know the specific type of lymphocyte that you're looking at and the percentage of which, you need to utilize cytochemical staining or flow cytometry to properly identify these lymphocytes. The only three morphological stages that we talk about when it comes to lymphocyte development is lymphoblasts, prolymphocytes, and lymphocytes. That is it. These developmental pathways are shared within these subpopulations, but again, the CD markers and the cell functions varies, but we cannot tell that apart visually. Lymphoblasts are by far the most immature distinguishable cell, it has this really high nucleus to chromatin ratio. See how big that cell is and how much of that cell is occupied by the nucleus? That is what we call a high um, nucleus to cytoplasm ratio. Also, I want to note the size of the cell as well. They are significantly bigger than these RBCs that they surround. We also talk a lot about chromatin patterns. This openness that you see in the nucleus, how much light is speaking through. You'll hear the terminology lacy and fine. To steal from a lab tech that I work with, um, she also likes to think of these as fuzzy. Also, the cytoplasm is usually very scant. You can't really see anything inside, usually very smooth. And it is also slightly blue or basophilic and does not have granules. Also, I want to note these clearings here. Those are called nucleoli. If you see them in high amounts within a cell, then that means it's more likely than not a blast. Pro-lymphocytes you can't really see too well in the bone marrow. Just think of it like a slightly larger version of a regular lymphocyte. They have a high NC ratio, but they're much more clumped and might have nucleoli. But they're not as scary looking as the lymphoblasts are. And again, there shouldn't be any granules inside the cytoplasm, and we're just looking at a slightly blue cytoplasm. Lymphocytes, and this is a very classic photo of a lymphocyte. They are usually on the smaller end, but they can be slightly larger than this. The nucleus, a lot of people like to say, especially in a small lymphocyte, is about the same size as a red cell, but I would argue that this lymphocyte is slightly larger than normal. Um, it does occupy a majority of the cell, but the thing that makes it not so scary and not like a blast is that NC ratio. Or not that NC ratio, how condensed that nucleus is compared to the lymphocyte. You can't really see anything through it. No visible nucleoli might be true. I've also have observed that some lymphocytes have what looks like a nucleoli, but it's not a true nucleoli. But it just appears that way to a lab tech. Again, they can be small or large. They have a pretty wide variety. Um, but if you notice these large lymphocytes that may have granules in them, those aren't really the same type of granules as a neutrophil is. They should only really make up a small percent, and they're not considered atypical lymphocytes. This is something that I think a lot of newer techs would want to do when they see a large lymphocyte like this. Um, they aren't usually activated. They are usually just natural killer or cytotoxic T cells if you see those granules with that huge um, lymphocyte, but they're not what we call activated or atypical lymphs. So these are cytochemical stainings that you'll need to know. This is how lymphocytes can. This is each of the common stains that you'll see in the pathology lab, and the results to help tell you whether or not you're looking at something that's from the myeloid line or the lymphoid line. And this is what this lymphoid line should looks like. If you prefer tables, this is a pretty nice slide, and that's what a large lymphocyte will look like at the bottom there. There are granules there, but they're not quite to the same level as a neutrophil is. And it's not activated either. There's no basophilia, and they're not doing true scalloping where they're trying to reach out and touch each of the cells. All right. B lymphocytes, they are created and mature within the bone marrow. The antigen independent maturation occurs within the bone marrow, but the dependent continues on into the lymph node and the spleen. Again, B lymphocytes make about 20 to 30% of your lymphocytes overall. And the CD marker expression helps tell you where the age of the B lymphocyte is. So CD19 is considered pan B. That means it's just always there. But have a high amount of CD10 cells. That means that you're looking at a possible acute lymphoblastic leukemia because they are only found during B lymphocyte development. Any again, high expression of CD10 found in the peripheral blood is associated with ALL, or again, acute lymphoblastic leukemia. So we are so like I mentioned, these CD markers and IgGs, they'll tend to appear and disappear as the cell develops. The earliest committed B cell precursor is called the pro B cell. It'll stain CD34 positive, CD19 positive, and CD10 positive. Next, it turns into a pre B cell or hematogone, which has either late or early stage. In early pre-cell development, it'll be CD4 negative, CD19 positive, CD10 positive, CD20 positive, and CD24. Late pre-cell is CD19, CD20, CD24, CD22 pos, and CD24 pos. Immature or mature B cells that are able to recognize self antigen with a high avidity to the point that it doesn't really want to let go will undergo either receptor editing or apoptosis. That way, you're not going to get autoreactive B cells, which is not going to be very good for you, especially if you're trying to avoid a autoimmune disease. B lymphocytes are incapable of binding antigen until both IgM and IgG are expressed on the cell surface. The naive lymphocytes are released from the bone marrow and into the peripheral blood, and then they can enter in the antigen dependent stages as they go into the lymph node in the spleen. When they enter their secondary lymphoid organs, they'll wait for their antigen. The the lymphoid organs will be filtering blood and sorting out the pathogens. Once the B cell finds the pathogen that matches their markers, then they're going to migrate to the medulla or the spleen. Upon that migration, they're going to develop into these B cells called plasma cells. Um, the plasma cells will then undergo clonal expansion. Their job is to produce B or T cells, or sorry, IgG or IgM that have a high specificity to the antigen. When they undergo that clonal expansion, that means they'll also be immortalized, which means they'll be a long-lived memory cell to help provide long-term immunity. That way you don't get sucker punched at the same infection again. So, binding when an antigen binds to a naive B cell, that's going to create the primary immune response, or the first time immune response. And the largest immunoglobulin in that response will be IgM, as in the largest amount. The second time that antigen binds, or binding to a memory B cell, that will create the secondary immune response, and that'll generate IgG at a high level. Those antibodies are functions to help neutralize and eliminate bacteria. That way, they serve as a strong signal being like, please eat these up to any cell that is able to eat them up. Some antibodies will also activate complement. That way, that it'll enhance phagocytosis or cell lysis. Again, plasma cells will produce the antibody based on the isotope of the heavy chain and the antigen that is specific to that antigen that activated the cells. Plasma cells only represent about less than 4% of the cells in the bone marrow and are never really seen in the peripheral cell in high amounts. Usually, you'll see them more often in the lymph nodes. A lot of these conditions are very strongly associated with plasma cells, and when they do happen, then that might increase plasma cell activation and migration. Any alterations to IgG production will lead into these specific disease states, and these are terminology that is good to get familiar with. Hypogammaglobulinemia means that you are not making any antibodies. Polyclonal gammopathy means that you have different Ig immunoglobulins expanding, Monoclonal means that there's only one immunoglobulin that is expanding. So these are the marker changes as a B cell matures. Um, if you are in rheumatology, immunology, then you will have to know these. If you are a lab tech, I would just sort of recommend glancing over these at a very general stance. B cells will recognize and interact with allogenic proteins using a specific B cell receptor. The B cell receptor is made up of a homodimer pair. Basically, they just combine two immunoglobulins together. BCR, so they'll be made up of two heavy chains and two light chains. After they encounter an antigen, then you might see the BCRs undergoing isotope switching, which is by replacing that constant region that is found within the heavy chain. That'll help it develop a different immunoglobulin. Again, immunoglobulins are proteins that are developed by your B cells and your plasma cells, and they are made up of two pairs of polypeptide chains. Too heavy and too light. Too heavy, depending on which heavy chain you're looking at, determines the antibody class. Too light is just another additional variation, adding to the complexity of immunoglobulins. One immunoglobulin will have one pair of heavy chains and one pair of light chain molecules, which will be identical. Each immunoglobulin contains different subclasses, and those subclasses have distinct physical and biological properties. The constant region will be the same for all antibodies of that class, and that is determined by the heavy chain. The variable region will differ, and that allows for the wide diversity of immunoglobulins that we see in order to help develop antigen specificity. And this is what that looks like. T cells are created in the bone marrow and mature into the thymus. In the thymus, any developing T cell that is found in the thymus are called thymocytes. T lymphocytes constitute about 70 to 80% of your peripheral blood lymphocytes, and 60% of those T cells are CD4 helper cells. The remaining 20 to 40% of those T lymphocytes are the CD8, which is cytotoxic CD8 T cells. The ratio of CD4s to CD8 cells should be 2 to 1, and that's important in order for your immune system to be able to function. You could see the decrease in this ratio being heavily demonstrated in something like in viral infections or immunodeficiencies like AIDS. You could also see increases in this, such as acute graft host versus host disease, scleroderma, and multiple sclerosis. T lymphocytes also possess these Fc receptors for IgGs, as well as the other CD markers. Again, good for you to know. The CD markers, just like B cells, will vary with expression based on where that cell is along the pathway. So TCRs are usually right next to the CD4 and CD8 markers. Their job is to help restrict or facilitate, recognize, and restrict TCR binding, depending on what the cell is. So CD4 T helper cells will recognize HLA class 2, and CD8 cells will recognize HLA class 1. By the way, when you see the terminology MCH molecules, that also means that also means HLA for all intensive purposes. Technically they're different, but for most lab techs it's basically the same for you. IL-7 and tumor necrosis factor alpha will help drive T cell differentiation from the CLP. CD1 is the first indicator that you have a cell that is committed to the T cell lineage. So T cell development is very complex. Um, when I say like something like double negative and double positive, um, that means it has either recognized two forms of the antigen, or it's able to positively identify them. So pro and pre thymocytes are developing in the thymus, and they're actually 95% of those pro and pre thymocytes are going to be eliminated because they haven't done their job just right. That two-step selection is really important to make sure that we're not going to get something that's going to attack our own body. So, in order to undergo positive and negative selection, we're talking about these two processes. So, positive selection means that you are making sure that the self and non self is able to be recognized, that it's able to weakly bind to these self antigens and also ensure that we're looking at the right class of um, of receptors. Negative means that we are trying to make sure that it will not bind to self antigens so strongly that it won't leave. If it's if it binds so strongly, then that cell will receive death signals and eventually die off. If it's unable to interact at all, then it will just cease to exist. Again, class 1 HLA will interact with CD8, class 2 will interact with CD4. They can only become effectors once they actually make contact with their antigen. So HLA just stands for the human leukocyte antigen system. Again, for all intents and purposes, HLA is the same as MHC. DHLA loci are found on chromosome 6 and are divided into three different classes here. HLA class 1 will be found on any nucleated cell and detect intracellular changes, which will then cause that expression of class 1 to become non-self. Class 2 will be found on antigen presenting cells, and their job is to detect extracellular proteins. And class 3 is just dedicated to complement. And this is sort of what the function of T lymphocytes look like. I've always found it really funny, this image. And again, this is what the HLA class system will look like. So T lymphocytes are really good at combating intracellular pathogens. Um, so T lymphocytes, sort of as a evolutionary check, cannot bind to their own antigen. The antigen must be presented to them. And again, they are MHC restricted. They can only interact with the appropriate MHC if they have that proper subtype. So in order to do that, they must possess both a functioning TCR and a co-receptor. And again, class 1 is associated with every nucleated cell and only interacts with CD8 T cells. And class 2 is found only on antigen presenting cells. And those antigen presenting cells will produce extracellular contents to show to CD4 T helper cells. So antigen presenting cells their job is to just show things to your lymphocytes. They also will help not only just presenting, but also secrete cytokines to tell the T cell to develop. The most effective antigen presenting cell is called the dendritic cell, and it's found in the skin, respiratory, and GI regions of the body. It's also a specialized macrophage with a lot of little cytoplasmic projections. Excuse me. Macrophages and B cells will also function as antigen presenting cells. There are four types of CD4 T helper cells. I'm not going to read these all out to you, but just understand that there are four different subtypes, and they produce specific pathogens, or they fight against specific pathogens, not produce specific pathogens, and with unique functions and roles. So CD8 cytotoxic T cells require antigen presentation for MHC class 1. They do acquire so what makes them different from CD4 cells is that their receptors are very good at targeting cells and killing off cells, with these cytoplasmic granules that are very good at destroying things. What happens is that a CD8 cell, once it finds its appropriate target, will release perforin. Perforin will then cut a hole into that cell membrane and then just dump all those toxic granules into the cell and trigger apoptosis. Memory T cells are just there to help trigger a rapid immune response after subsequent antigen exposure, and they're able to stay and lie dormant for years. So they'll live in G0 where they're just sort of continue living. They're not exactly dividing, they're not exactly replicating. They're just living. Once the antigen is presented though, then they're going to produce a large and fast and more efficient response than the naive T cells will be. Natural killer cells. They are, again, part of the innate immune response, not the adaptive. They only are found in, they will develop and mature just in the bone marrow, unlike the other two. And they make up about 5 to 15% of all lymphocytes in your peripheral blood. The natural killer cells are different in terms of CD markers from your T and B lymphocytes. So CD58 and CD16 will be positive, and they'll have a bit of weak reactivity with CD8 and CD12. Natural killer cells they don't really need to rearrange their gene receptors if they don't really good can't really combine with stuff, but instead what they have is KIR or killer immunoglobulin receptors that are able to interact with a huge group of ligands. They are not restricted by MHC and they do not interact with antigen presenting cells. And this is sort of their general process with the CD markers that come and go. So activated or atypical lymphs just mean that there is a lymphocyte that is reacting to something. So immunocompetent and naive cells just mean that they haven't been able to find their antigen just yet. But once they find the antigen that they're able to bind to, then they'll undergo clonal expansion. The activated B and T cells actually become larger and become mitotically active again, and they develop into these immunoblasts. Eventually, those immunoblasts will develop into these four types of activated lymphocytes. So reactive lymphocytes can either be B or T lymphocytes, and they will demonstrate at least one of these characteristics. They'll have a much larger cell size and a decreased NC ratio. The nucleus will start forming really weird shapes. So most lymphocytes have this neat little oval. A lot of them just start stretching and elongating, and they kind of do whatever they want. The chromatin just becomes much lighter than it was originally uh as a as a inactivated lymphocyte. You'll also see a lot of basophilia, especially concentrated where it's interacting with other cells. You can also see some of these granules in there, maybe vacuoles, depending on the condition of the patient. We also see a lot of this cytoplasm which will like start to wrap around red cells. That's called indentation or scalloping. We don't really see atypical or reactive lymphs in high numbers in healthy individuals, but if we see them in high amounts, then we're thinking viral infection. Immunoblasts, I'm not going to just describe again, but they look like blasts. Again, these immunoblasts will either become B cells or T cells, which will then continue down their further pathways. But you're not going to see immunoblasts during viral infections. Plasmacytoids are kind of weird. Um, They look like a regular B cell, but their cytoplasm will show that they're expressing like a plasma cell. They might have a nucleolus, but the nucleus tends to be in the center of the cytoplasm, unlike a plasma cell, but it'll usually look very deeply blue. You don't really see them in the peripheral blood during viral infections. Plasma cells are very unique. They um they're become very large and become slightly oval. The nucleus is often off-center eccentric and contains like this like cartwheel or bicycle spoke like arrangement within the nucleus. They also have a really big Golgi apparatus, so you're gonna see this like clearing in the cytoplasm surrounded by a very deep blue cytoplasm. They're not likely to be present normally, but you can see them. So these are all really good examples of each of these cells. Reactive lymphocytes just kind of they don't have to check off all the characteristics that I described, but if they're demonstrating a good amount and don't look like a regular lymphocyte, then you're probably looking at a reactive lymphocyte. And that bottom one there is a really good example of a plasma cell. Memory cells are activated lymphocytes that can live for months and years. Both T and lymphocytes can again develop into memory cells, but you can't really tell them apart from just non-activated lymphocytes. Again, their job is to help elicit a second, when you have a second immune response, and it's gonna be much more fast and much more powerful than your primary, which means there's less lag time, more IgG with a longer plateau. That's the end of that lecture.